Split hand phenomenon in motor neuron disease, ALS. The split hand phenomenon is one of the most characteristic clinical signs of amyotrophic lateral sclerosis, ALS, the most common form of motor neuron disease. It refers to the disproportionate wasting of the thenar muscles and the first dorsal interosseous muscle, with relative preservation of the hypothenar muscles. In other words, the muscles on the thumb side of the hand become markedly wasted, while the muscles on the little finger side remain comparatively preserved, especially during the early stages of the disease. This selective pattern of muscle wasting gives the hand a distinctive appearance and serves as an important bedside clue in diagnosing ALS. The muscles most commonly affected include the abductor pollicis brevis, APB, opponens pollicis, flexor pollicis brevis, and the first dorsal interosseous, FDI. These muscles gradually become weak and atrophic, leading to difficulty with thumb abduction, thumb opposition, precision grip, and fine finger movements. In contrast, the abductor digiti minimi, ADM, and the other hypothenar muscles are relatively preserved in the early phase of the disease. Clinically, this results in a flattened thenar eminence, wasting in the first web space between the thumb and index finger, visible fasciculations, weak pinch grip, and difficulty performing everyday activities such as writing, buttoning clothes, turning a key, or holding small objects. One of the most frequently asked questions is why the thenar muscles are affected much more than the hypothenar muscles. The exact mechanism has not been completely established, but several complementary theories explain this selective vulnerability. The most widely accepted explanation is the greater corticomotoneuronal or corticospinal input to the thenar muscles. The thumb muscles, especially the abductor pollicis brevis and the first dorsal interosseous, receive dense direct projections from the primary motor cortex. Since ALS preferentially affects these corticospinal pathways, muscles that depend heavily on cortical control undergo degeneration much earlier. This concept is considered the cornerstone of the split hand phenomenon and is the explanation most commonly expected in examinations. Another important factor is the specialized function of the thumb in fine motor control. The thumb is involved in highly skilled movements, such as writing, buttoning clothes, picking up small objects, using a smartphone, typing, and performing precision pinch. These activities require continuous activation of the thenar muscles throughout daily life. Because these muscles are used so frequently, they experience greater metabolic stress and neuronal workload over many years. This increased functional demand is believed to make their motor neurons more susceptible to degeneration in ALS. The higher metabolic demand of the thenar muscles is another proposed mechanism. Motor neurons supplying these muscles require greater amounts of ATP, increased mitochondrial activity, and higher oxygen consumption to maintain their continuous function. ALS is known to cause mitochondrial dysfunction and impaired cellular energy production. Consequently, neurons with higher metabolic requirements are more likely to fail earlier, resulting in preferential degeneration of the thenar muscles. Another important mechanism is glutamate excitotoxicity, which is a well-recognized pathological process in ALS. Glutamate is the principal excitatory neurotransmitter in the central nervous system. Excessive glutamate activity causes increased calcium entry into motor neurons, leading to oxidative injury, mitochondrial dysfunction, and ultimately programmed cell death. Experimental evidence suggests that the motor neurons supplying the thenar muscles and the first dorsal interosseous are particularly susceptible to this excitotoxic damage. Cortical hyperexcitability also plays a significant role. One of the earliest physiological abnormalities detected in ALS is increased excitability of the motor cortex. This excessive cortical activity predominantly affects motor neurons projecting to muscles with extensive cortical representation, particularly the thumb muscles and the first dorsal interosseous. As a result, these muscles undergo earlier degeneration than muscles receiving less cortical input. Motor neurons themselves are not biologically identical, and this forms another explanation for the split hand phenomenon. Motor neurons supplying the thenar muscles differ from those supplying the hypothenar muscles in terms of gene expression, ion channels, neurotransmitter receptors, protein metabolism, and other intrinsic cellular properties. These biological differences make certain motor neuron populations inherently more vulnerable to the neurodegenerative process of ALS. Abnormal calcium handling has also been implicated. Healthy motor neurons regulate intracellular calcium very efficiently. In ALS, motor neurons supplying the thenar muscles appear to have a reduced capacity to buffer intracellular calcium. Excess calcium accumulation activates multiple destructive cellular pathways, ultimately leading to neuronal injury and death. This impaired calcium buffering further contributes to the preferential involvement of the thenar muscles. Oxidative stress represents another major pathogenic mechanism. ALS is associated with excessive production of free radicals and reactive oxygen species, which damage proteins, lipids, mitochondria, and DNA. Because thenar motor neurons have greater metabolic activity, they experience higher oxidative stress and therefore undergo earlier degeneration compared with other motor neuron populations. The accumulation of abnormal proteins also contributes to selective neuronal degeneration. ALS is characterized by pathological aggregation of proteins such as TDP43, SOD1 in familial ALS, FUS, and abnormalities related to the C9 or 72 gene. Certain motor neuron pools appear to be more susceptible to the toxic effects of these protein aggregates, resulting in earlier degeneration of the neurons supplying the thenar muscles. A natural question then arises, why are the hypothenar muscles relatively spared? The first explanation is that the abductor digiti minimi receives fewer direct corticospinal projections compared with the thenar muscles. Because these neurons depend less on cortical input, they are relatively protected from the corticospinal degeneration characteristic of ALS. The hypothenar muscles are also less involved in fine motor activities. Unlike the thumb, which is essential for precision grip and dexterity, the little finger mainly contributes to power grip and stabilization of the hand. Consequently, the motor neurons supplying the hypothenar muscles experience lower functional workload and reduced metabolic stress throughout life. Another contributing factor is their lower metabolic demand. Since these muscles require less ATP production and reduced mitochondrial activity, they are less vulnerable to mitochondrial dysfunction and energy failure, allowing them to survive longer during the disease process. Hypothenar motor neurons may also possess better calcium buffering capacity, allowing them to regulate intracellular calcium more effectively. This protects them from excitotoxic injury and delays neuronal degeneration. Similarly, they may have greater resistance to oxidative stress, stronger antioxidant defenses, and more efficient mitochondrial reserve, all of which contribute to their relative preservation. Furthermore, the motor neurons supplying the hypothenar muscles may express different protective proteins and survival promoting factors, making them intrinsically more resistant to ALS pathology. They may also experience less glutamate-mediated excitotoxicity, reducing calcium influx and limiting neuronal injury. An important examination question is why ALS does not affect both thenar and hypothenar muscles equally, since both are supplied by lower motor neurons located in the cervical spinal cord. The answer is that although these motor neurons are anatomically close to one another, they are biologically distinct. They differ in their cortical connectivity, metabolic activity, gene expression, mitochondrial function, calcium regulation, oxidative stress resistance, glutamate sensitivity, and protein handling. ALS is therefore not a disease that damages all motor neurons uniformly. Instead, it is a disease of selective motor neuron vulnerability, in which some motor neuron populations degenerate much earlier than others. The first dorsal interosseous muscle deserves special mention because it is also preferentially affected despite not being a thenar muscle. This is because the first dorsal interosseous receives strong corticospinal input, has a large cortical representation, and participates extensively in fine motor movements. Consequently, it behaves similarly to the thenar muscles and undergoes early degeneration in ALS. From a clinical perspective, the split hand phenomenon is extremely valuable. It is considered one of the characteristic bedside signs of ALS and helps differentiate ALS from disorders such as cervical radiculopathy, ulnar neuropathy, median neuropathy, and many peripheral neuropathies, where this selective pattern of muscle wasting is generally absent. The split hand can also be quantified electrophysiologically using the split hand index, SHI, during nerve conduction studies, providing additional objective support for the diagnosis. In summary, the split hand phenomenon is characterized by preferential wasting of the abductor pollicis brevis and the first dorsal interosseous muscles, with relative preservation of the abductor digiti minimi. The most widely accepted explanation is the selective degeneration of corticomotoneuronal pathways supplying the thenar muscles. Additional contributing mechanisms include fine motor specialization, higher metabolic demand, glutamate excitotoxicity, cortical hyperexcitability, mitochondrial dysfunction, oxidative stress, impaired calcium buffering, intrinsic motor neuron vulnerability, and abnormal protein aggregation. The hypothenar muscles are relatively spared because they receive fewer corticospinal projections, have lower metabolic demands, perform fewer precision movements, and possess biological characteristics that provide greater resistance to degeneration. Thus, the split hand phenomenon beautifully illustrates a fundamental principle of ALS. The disease is one of selective motor neuron vulnerability rather than uniform degeneration of all motor neurons. If you'd like, I can also convert this into